I think we should get started. For some reason, this walking mic is not working, so I'll have to stand at the podium a little bit more. As usual, I'll do what I can to get the grades posted as soon as possible. I expect no later than Thursday or Friday, right? So maybe even sooner, depending on how fast the scanning office works, okay? So just a reminder, next week, the quiz 3 and 4 are due. That's the hyperlipidemia and the hypertension quizzes, which correspond to the hyperlipidemia, hypertension lectures that have been on YouTube for the last few weeks, okay? So just a reminder, that's due next week. So today what we're going to do is we're going to start the cancer section. Okay. And in this section, what we're really trying to get at is understanding cancer as it sort of originates in the body and as it behaves biologically. Okay. Really fundamentally, what is cancer? So cancer is really a disruption of the normal controls that are put on cellular societies in our body. Okay? So a cancer is really any abnormality in the control over things like cell growth, cell differentiation, and cell division. Okay? So cells in our body, they all belong to cell societies, right? They all live in tissues, which are part of organs. And these cell societies, there's nothing democratic, right, about the cell societies in our body. They are highly controlled. It's very much a fascist state in our body. So cells in our body are told, really, when their existence is desirable, okay? And in fact, they actually require constant reassurance, right, in order to get the signal to continue to function. Okay? And only under very specific conditions are cells ever allowed to grow, right, to grow in size or to grow in number, right, to divide. And tissue types are also all, they also exist in different differentiation steps. Okay, so all of our epithelial tissue, for example, are derived from adult line stem cells, right? So they all start off as stem cells, and they go through differentiation steps, and those differentiation steps are also an important part of maintaining a normal cell society. Okay, so the cells in your body are under a tremendous amount of control. Okay? And all of those controls are coded in your genome through a set of genes that we call social control genes. Okay? It's a very, very diverse array of genes, but really they are any gene that codes for a protein that is some way involved in cell growth, division, and differentiation. And cancer is a disruption of those genes really at the genetic level. It is a mutation in those genes. Okay. So social control genes, they do things like direct, stimulate, inhibit, or regulate, as I said, things like cell division, cell differentiation, cell growth, DNA repair, okay, which is, of course, part of social control, cell mortality. And when we talk about cellular mortality, it's different than we think of our own mortality. Okay? Cellular mortality is really a description of the number of times a cell is allowed to divide itself. Okay? There's a limit to how many divisions a cell can do. Okay? So it's really, when we talk about cell mortality, it's really the mortality of that specific set of genes of that cell. Okay? And then social control genes also govern apoptosis. that programmed cell death. Okay? Because every cell in our body has that suicide program, has that apoptosis program, and it's absolutely necessary for them to be able to respond to signals to trigger apoptosis. Okay? So these are our social control genes, and cancer fundamentally begins with something that we call the transformation, right? The transformation of a cell, which is basically a mutation in one of these social control genes. Okay? So the phases of the pathogenesis of cancer, the first stage is transformation, where a normal host cell is transformed into a malignant cell. Okay? And of course, not every mutation in one of these social control genes is going to produce cancer. It has to be a mutation that results in unregulated growth, division, differentiation, okay, or any one of those other sort of social control mechanisms. So the outcome of the mutation is this unregulated and extensive growth division of that cell. So the next phase is the growth of the transformed cell. Okay, so it's not enough that just one cell becomes transformed. You actually have to have the proliferation of those cells. Okay? And usually that proliferation develops into a tumor. Okay? So that cell now forms a tumor. And if you look at the cells within a tumor, what you'll find is that they're all genetically identical, pretty much, because it's a single cell that's become transformed, and now it's making lots and lots of copies of itself. Okay? And a good thing to keep in mind is that, you know, we think of cancer as being this like catastrophic thing that happens very rarely. But I can tell you that transformation of cells happens all the time in your body. Okay? Transformation of cells in your body is not an uncommon event. Many of the cells that that accumulate mutations, they accumulate mutations that do not result in survival. Okay? And so that causes the cell to die. Or it accumulates a mutation, and then that triggers the immune system around it to kill the cell. Okay? So transformed cells, the transformation of cells is not an uncommon thing, but the continued growth, right? The continued growth of that transformed cell is fairly uncommon. Okay? That's really where cancer begins. And then once you have that tumor, the next stage is that tumor gets bigger and it starts to invade surrounding normal tissue. Okay? So local invasion, and usually what's part of that local invasion is a process called angiogenesis, which is the formation of new blood cells that directly supply blood into the tumor. Okay? So it's like the cancer starts developing this little town, right? And then that town gets bigger and bigger, and now it's a city, and part of becoming a city is constructing its own major highway. Okay? So creating its own blood supply is a key part of the growth of a tumor. Now, when we think about local invasion into surrounding tissue, cancer is not nice. It does not just push aside what's surrounding it, right? It doesn't displace surrounding tissue. It eats a hole through whatever it encounters. Okay? So it's literally going to eat its way around this local tissue. And the next step, which doesn't always happen with every cancer type, but this tumor is going to get larger and larger. It's going to invade locally, and it may end up metastasizing into another organ. And that metastasis can either be through what we call direct extension, meaning that it's the same tumor that started in one organ and then just has now grown into a neighboring organ. Okay? Or you can have what we call distant metastasis, and that's on this slide, that's the last stage. And that's where cells of the primary tumor, of the original tumor, break away and travel either through the lymphatic system or through the bloodstream, and they colonize a distant site. Okay? And so, and all the sites of metastasis are secondary tumors. There's a primary location, and then they form distant secondary locations. And not every cancer forms a distant metastasis. Okay? Different cancers behave differently, and the best predictor of the behavior of a cancer is the cell type of origin, basically the organ of origin. Okay, breast cancers tend to spread in a stereotypic fashion. Colon cancers tend to spread in a stereotypic fashion. Okay, prostate cancer, so forth. Okay. Endometrial cancer tends to never leave the uterus, right? It doesn't tend to actually form metastases at all. Okay? So we'll talk a little bit more about that later, as the different behaviors, different patterns of spread for different cancer types. That's the basic phases of pathogenesis and spread. So fundamentally, this is a disease process where you have altered cell growth, cell division, or differentiation. Okay? So let's just hit on some of sort of the key aspects of normal cell division, growth, differentiation. Okay? And then talk about where things sometimes go wrong. So when we talk about cell division, okay, cells in our body undergo a process called mitosis. Okay? Cellular division for most cells in the body occurs through a process called mitosis. And through mitosis, cells make an identical copy of themselves. Okay? Now, the cell cycle describes basically the entire cycle between a cell that is not dividing all the way up until it's completed a cell division. Okay? So cells start in a phase of the cell cycle that we call the G1 phase. Okay? And most cells in your body are not actively dividing. Okay? And so most cells in your body are in that G1 phase. Okay? So in this phase, you get DNA replication going on. The cell is making a copy of its DNA. Okay. Then it's getting ready for DNA replication. I apologize. It's getting ready for DNA replication. Then it moves into the S phase, or the synthesis phase, where it actually replicates DNA, actively replicates the DNA. And then the G2 phase, it's making a copy of its intracellular material. Okay? It's making duplicates of all of its intracellular proteins. And it's that final phase for getting ready for cellular division. Okay? And the cellular division is the M phase, or the mitosis phase. Okay? And technically, the mitosis phase is actually nuclear division. Okay? But at the tail end of nuclear division, you actually get the total cell division. Okay? So G1, getting ready for DNA replication, S phase, active DNA replication, G2, we're replicating everything else. The M phase, we're dividing the nucleus, and at the tail end of that, we're dividing everything else. We're producing 2 cells. Okay? And probably haven't looked at this in many years, but this is the phases of mitosis, right? You probably first saw this in the 4th grade, and then again periodically throughout your young adult life. So the mitosis phase, of course, involves the lining up of chromosomes, and then the splitting away of duplicate chromosomes, and then at the tail end, cellular division. Okay? And I put this up simply to illustrate to you that you rarely see, when you're looking at cells histologically, when you're looking at a tissue sample of normal tissue, you rarely see cells in any phase of mitosis. Okay? Because only cells that are actively dividing, only tissue with actively dividing cells will ever be in any stage of mitosis, right? Most cells are in that G1 phase, right, which is very far away from the M phase. However, when you look at tumor cells, when you look at malignant cells, you will often see cells in the middle of the most recognizable phases of mitosis. You will often see cells in metaphase, where the chromosomes are lined up, cells in anaphase, where the chromosomes are being pulled at opposite ends of the cell. Okay? So this is one of these sort of visual markers when looking at malignant cells that you will often see. And it's also part of, you can actually quantify, based on what the cellular density that you're looking at, the number of cells in active division, and that's part of an assessment of how aggressive the tumor is, right? The rate of cell division, it goes into assessing how aggressive this cancer could be. Okay? Now, at every stage of the cell cycle, G1, S, G2, there are a set of proteins in the cell called cell cycle inhibitors. Okay? And at each stage, there's a whole set of cell cycle inhibitors that prevent the cell from moving on to the next phase. Okay? Some of these proteins might bind to portions of DNA, preventing them from being replicated, okay? Or preventing them from being packaged in such a way that they can move on to the next phase. At every stage of the cell cycle, in order for the cell to move forward, these cell cycle inhibitors have to be overcome. Okay? So this is one of these insurance policies to prevent the cell from inappropriately dividing. Okay? The default is don't divide, and we're going to put brakes at every turn. Okay? So that if we really want the cells to divide, we have to give it a very specific set of signals to allow it to divide. Okay? And what you'll find is some of these cell cycle inhibitors, okay, some of the really powerful ones, are very common genes that are mutated in cancer. Okay, so that all of a sudden those proteins don't work. And so you don't get that tonic inhibition. Okay? So cell cycle inhibitors are a very important target for potential mutation and transformation of a cell. Now, cells that we think of as being non-dividing cells, right? So neuronal cells in our body, myocytes of our heart, so the muscle cells in our heart, all of these cells are non-dividing because they are strongly frozen in a stage of the cell cycle called the G sub 0, which is similar to the G sub 1. Okay, but it generally does not allow the cell to progress under any circumstances into further stages of the cell cycle. So for those non-dividing cells, you don't see cancers that originate as that tissue. Okay? So for example, for primary brain tumors, they are never of neuronal origin. Okay? In fact, all of the primary brain tumors originally are glial, so they're one of these non-neuronal supportive cells in the brain. Okay? So you might have heard technical names for primary brain tumors as a glioma or astrocytoma, right? So these are all glial cell names, non-neuronal. Because they retain the ability to divide, but the neuronal cells can't divide, so they can't get transformed. The other thing to mention before I move away from the cell cycle is anytime you have cellular division, under normal circumstances, anytime you have cell division, cell turnover, you increase the risk, or there is a risk, for mutation leading to malignancy. Okay? So anytime you have cell division, there's a chance for mutation and a chance for transformation. So there are a whole host of DNA repair proteins, okay, or DNA reading proteins that are designed to identify potential mutations. Okay? And what they do is they identify the potential mutation. They sometimes can repair it themselves. And if they detect too many errors, they will trigger apoptosis of the cell. Okay? So that the mutation doesn't reproduce, essentially. Okay? So these DNA repair proteins are also a target for potential mutation leading to the development of cancer cells. Okay? If you've ever heard of the BRCA1, BRCA2 genes that increase dramatically the lifetime risk for breast cancer in women, BRCA1, BRCA2 are DNA repair proteins. Okay? And actually, every man and woman is born with 2 copies of BRCA, BRCA1 and BRCA2. Okay? And having a mutation in one of those pairs dramatically increases your lifetime risk of all cancer. But for some reason in women, it particularly increases the risk of breast cancer, uterine cancer, ovarian cancer. Okay? But at the heart of it, it's really about the loss of DNA repair. Okay. And then there's cell differentiation. Okay? And we talked about cell differentiation in the context of the immune system, because we talked about how all of the white blood cells, in fact all the white and red blood cells, are derived from the same adult stem cell line, the hematopoietic pluripotent stem cell. Right? Now, there are certain tissue beds in our body where the cells are constantly renewed, like blood-borne cells, like epithelial tissue. Okay? The cells are constantly renewed. So you have a tissue line stem cell in the area that's constantly producing the fully differentiated cell type. Okay? So the process of differentiation starts with, if we start here, the pluripotent cell. This is a colony-forming cell, okay? And it undergoes differentiation steps that narrow the fate of the cell. Okay? At the top here, what we actually have is the totipotent cell, which was basically what we were when we were a little bit more than a twinkle in our parents' eye. We were in the afterglow of our parents' eye. This is the fertilized egg, right? So when the sperm and the ova fused, they produced a single cell, right, of which all other cells that make you came from. So that's why we call it the totipotent cell. It was our original stem cell. And then that totipotent cell undergoes a wide variety of differentiation steps to produce all of you. Okay? But then at the adult stem cell line, we have lots of colony-forming stem cells. Okay? Now, you can have several differentiation steps between the colony-forming stem cell or the tissue stem cell and what's called the terminally differentiated stem cell. Okay? You can have multiple differentiation steps. Now, with every differentiation step, a few things happen. The fate of the cell narrows, and the mitotic activity starts to decline. Okay? So with every differentiation step, the fate of the cell narrows and the mitotic activity declines. Okay? So that when you finally get to the terminally differentiated cell, now you have a cell that is the most functionally mature, and you also have a cell that has lost the ability to divide. Okay? Now, you think about, we talked in the first section about the metaplasia in the airways as a result of chronic smoking. So the fully differentiated cell type in our airways are those ciliated columnar cells, right? Now, if you are a chronic smoker, sometimes those cells will not look ciliated and columnar. All of a sudden, they'll look like simple squamous cells, right? Because the simple squamous cells can handle the constant injury because they're very mitotically active. They're less differentiated, and they're very mitotically active, right? That was the metaplasia. But you lost the function of the epithelial tissue. So now you don't have that beautiful cilia that helps you move that mucus out of your airways. So you wake up every morning with this thick mucus on your airways, and you have to cough for like 5 minutes before you can start your day, right? Because you got to physically remove all that mucus that's been building up throughout the night. Okay? Not that I've ever experienced this personally. I have. I was a former smoker, so I know this long, long time ago. So the terminally differentiated cell type is functionally mature, but it has an expiration date on it. It's going to die, and it's not going to divide. It's going to be replaced by a newly differentiated cell. So in the context of cancer, cancer cells often will refuse to differentiate, will resist differentiation. And in fact, the less differentiated the tumor cells are, the more aggressive the cancer tends to be, because it is very mitotically active. And it also contributes the least to the function of the tissue and potentially creates more problems for the patient. Okay? In cancers of blood-formed cells in leukemias, right? When you have leukemias that end up being highly undifferentiated, you get an overproliferation of highly undifferentiated cells. You end up with real loss, significant loss of immune function. You often have significant loss of normal red blood cell function. So patients can be highly anemic. They can be extremely immune deficient, right? Even though their white blood cell count may be through the roof. Okay? But all of those cells are malignant cells that they're undifferentiated and they don't contribute to the function of the tissue anymore. Okay? So what we find sometimes with cancers is that normally stem cells will undergo mitotic divisions and then differentiation steps, okay? Because you don't want to run out of stem cells. And so normally the stem cells divide, and then half of the daughter cells go on to differentiate. Okay? But when more than half of the stem cells remain stem cells, that's a pattern of potential tumor formation. Okay. When in a tissue bed, so over here this is actually, you have some villi, this is probably small intestine, right? And then the brush border cells of these villi originate from the stem cells that are in the crypts here. Okay? And so if half of the stem cells that divide, more than half of the daughter cells remain undifferentiated, refuse to differentiate, you start to see an accumulation of undifferentiated cells, and potentially that's the beginning of potential tumor formation. Okay. So high rate of cell division that's unregulated, and a resistance to differentiation. These are sort of characteristics of malignant cells. So I described before that cancer is essentially, it starts with the mutation of some social control gene. Okay? It starts with the mutation of a social control gene. And the resultant gene after the mutation, the gene that causes a cancerous pattern of growth, we call those genes oncogenes. Okay? Oncogenes, cancer-causing genes. Okay? And these oncogenes were originally a normal gene, but they've acquired a mutation that has turned them into an oncogene. Okay? Now there are two ways to produce an oncogene, and it really depends on what the normal function of the original gene was. You can turn on a gene to produce an oncogene, or shut off a gene to produce an oncogene. Okay? So for example, we have a whole set of genes in this group of social control genes that we call tumor suppressor genes. And tumor suppressor genes, their job is to limit cell division, to limit growth, to promote differentiation, to promote DNA repair. Any gene whose function contributes to preventing tumor formation is a tumor suppressor gene. Okay, BRCA1, BRCA2, they're originally tumor suppressor genes. Okay? So what can we do to that kind of gene to turn it into an oncogene? What do we do to that gene? We shut it off, right? If its function is to prevent tumor formation, if we shut it off, then we get the formation of tumors. That make sense? So, oncogenes that were originally sort of inhibitory genes that sort of inhibit cell growth, division, promote differentiation, their mutation is going to be something that causes the silencing of that gene or the inactivity of the gene. Okay. On the other end of the spectrum, we have a whole host of genes whose normal function is to promote cell division, to promote growth. Okay. These are things like growth factors. Okay. Receptors that respond to growth factors. Okay. A variety of cytokines stimulate cell division. These are all the genes that code for these proteins are all, what we call them are proto-oncogenes. Okay? They are sort of stimulatory for the cell division and growth process. The mutation that they acquire that produces an oncogene is a hyperactivity of the gene. Okay? Something that turns that gene on in a persistent way. Okay? Results in the overexpression of the genes. Okay? So you can see that oncogenes have two potential pools of sources. The proto-oncogenes that get activated and the tumor suppressor genes that get shut off. Nearly every hereditary form of cancer, can be linked to a tumor suppressor gene. Okay? It's a phenomenon that's not well understood why, but for some reason hereditary cancers tend more to be mutations in tumor suppressor genes rather than the proto-oncogenes. So keeping that in mind, keeping that idea of there being a pool of proto-oncogenes and tumor suppressor genes, there are then what we call two different mutagenic roots, right? Two roots of mutations, or two types of mutations that lead to the production of oncogenes and the manifestation of cancer. And it's sometimes called the stimulatory route and the inhibitory route, but it's basically that turning on a gene or turning off a gene. So in this slide, what we have is actually examples of targets or proteins whose genes could be originally proto-oncogenes or tumor suppressor genes. So on the left side of the slide, the purple side, are the proto-oncogenes, okay, the stimulatory pathway. So we've got at the top here, we've got growth factors, and then over here we've got receptors for growth factors. And then inside the cell we have intracellular signaling proteins that the growth factors work through. And then down here we've got transcription factors that the growth factors work through. Okay? All of these are potential targets, right? They're all potential proto-oncogenes, or the products of proto-oncogenes. And then in the inhibitory pathway, in here we've got a cell cycle inhibitor or potentially a cell differentiation stimulator. Okay? It stimulates the differentiation of the cell. Okay? Here we've got the receptors that those signals work through. Again, we have the intracellular signaling proteins that they work through and the transcription factors that they stimulate. Okay? So you can see that these tumor suppressor genes, proto-oncogenes, can really, there's a wide variety of potential targets for both of them. Okay? So any aspect under social control is therefore a possible mechanism for cancer disruption. So you saw cell signaling, right? So there's signaling pathways for either stimulation of growth or inhibition of growth. They are potential targets. Any of the cell cycle control, right? Any of those cell cycle inhibitors or cell cycle stimulators are potential targets. Any of the DNA repair proteins, anything that triggers cell growth, triggers cell differentiation. Transcriptional regulation. Is everybody familiar with what a transcription factor is? They are proteins that can control whether a gene gets expressed or not, right? So they are at the level, they're inside of the nucleus, they're at the level of DNA, and they bind to the beginning areas of a gene called a promoter region, and they determine whether a gene gets transcribed or not. Okay? So that will literally determine, right, whether that gene is overactivated or not activated at all. Okay? Senescence. Senescence. Senescence is all about cell mortality. Senescence is really the limit that any cell in our body has to the number of cell divisions it can undergo. And usually that's about 50 or 60. So a cell can divide about 50 to 60 times until that entire cell line can no longer divide. Okay? When a cell continues to divide beyond that point, we tend to describe that as being an immortalized cell. Okay? They are capable of passing on their genome in an unlimited way. And then, of course, apoptosis, this control over programmed cell death. Okay? So these are just some of the aspects of cell function that are under social control and therefore could be targets for cancer disruption. Okay? And these are actually real-life examples, identified examples of the products of proto-oncogenes and some proto-oncogenes and some tumor suppressor genes. Okay? So there's a variety of growth factors that have been identified as the products of proto-oncogenes, okay, where their mutations have been linked to certain cancers. There's a variety of tumor suppressor genes. And you see over here is the BRCA1, BRCA2, the DNA repair is one of those examples. There's two here that are incredibly important examples of highly common tumor suppressor genes, P53 and the RB protein. So both are cell cycle inhibitors. Okay? The P53 protein is a major cell cycle inhibitor. Okay? It's coded for by the P53 gene. And the expression of p53 also increases as the number of errors on DNA are identified by the cell. Yeah. So the expression of p53 actually goes up as the cell identifies errors on its DNA. Okay. So it's the break or the inhibition on the cell cycle gets stronger and stronger as the cell notices that there are more errors on DNA. And then once the amount of p53 hits a critical point, it triggers apoptosis. Okay. So once the expression of p53 hits a critical point, it just triggers apoptosis. Okay. And it's a way to prevent this, this cell that has accumulated too many errors, right? This highly fragile genome. It's preventing it from possibly passing on or promoting this these errors. Okay. So p53 is obviously a very important tumor suppressor gene, and what you find is that in cancers, often there are more than one mutation that you see in a cancer, and nearly half of all tumors of all types have mutations in p53. Okay. So nearly half of all tumors of all cancer types have mutations in p53. The RB protein is a very significant cell cycle inhibitor, okay? And it's coded for by the RB gene. RB stands for retinoblastoma because the mutation in the RB gene was first identified in that particular cancer type, retinoblastoma. And it is also a very common tumor suppressor gene for all types of tumors. No, excuse me, it's a common mutation for all types of tumors. So RB and p53 are particularly important tumor suppressor genes that are very common mutations occur in or many cancers commonly have mutations in those genes. Okay. So building up to this description, based on everything that I've described, I've told you so far, I'm going to describe to you some basic properties and behaviors of cancer cells. Okay? And I'm going to read this to you, and I want you to take this in. So, number one, cancer cells have the tendency to proliferate despite the lack of growth-initiating signals from the environment. Okay? So cancer cells grow and divide whether or not they have a signal or not. Okay? That is almost textbook definition of what we call autonomy. That first one is classic autonomy. The ability to replicate and grow without outside signals. The second bullet on here, cancer cells escape signals to die and achieve a kind of immortality in that they are capable of unlimited replication. Okay? So where a normal cell would trigger apoptosis and die, cancer cells are able to ignore those signals and continue to replicate. okay, and continue to pass on all of their mutations to their daughter cells. Okay? So they've achieved a kind of immortality. Their mutations, their genome has achieved an immortality. The third bullet, cancer cells lose their differentiated features and contribute poorly or not at all to the function of their tissue. Okay? And this is where you may start to see some signs and symptoms of the cancer besides simply a mass. Okay? Depending on what tissue is affected and how extensively it's affected, these cancer cells are not contributing to function. So you may see a loss of function, right, depending on the cancer type. Yes, ma'am. Let me get through this before, and then I'll go back. That's no problem. That's no problem. I thought you were asking about this one. So they lose their differentiated features, and they're not contributing to the function of the tissue. The fourth bullet, and this is an important one. Okay? They're all important, but this is a particularly interesting one that you may not have considered before. Cancer cells are genetically unstable, okay? And they evolve by accumulating new mutations at a faster rate than normal cells. Okay? So because these cells are constantly dividing, they tend to accumulate mutations. And so they can actually grow in their aggressiveness, potentially. Okay? And so you can end up with tumors that are populated by cells with multiple oncogenes. Okay? And they're evolving to try to continue to survive and continue to divide and multiply. Okay? And then finally, cancer cells invade their local tissue and overrun their neighbors. Okay? Cancer cell, this is how it spreads. They invade local tissue, they invade neighboring tissue, they can form sometimes colonies in distant sites, right, that we call distant metastases. So autonomy, this ability to basically continue to divide and grow without the need for any outside signal. Anaplasia is a term that specifically describes the loss of differentiation, or the lack, rather, of differentiation. Okay. So autonomy, anaplasia, two important terms to describe the behavior of cancer. Okay. Yes? Yes. Well, they do. They do. So whenever you lose the function of any tumor suppressor gene, you create a vulnerability. You don't create a definite formation of cancer. So that's why with like the BRCA mutations, a BRCA1, BRCA2 mutation means that it increases your lifetime risk. It doesn't mean that you're constantly forming cancer cells all the time, right? But it just creates a vulnerability that it can increase the likelihood. And then you add on top of that you know, environmentally we're always adding additional vulnerabilities by interacting with mutagens in our diet, in our environment. It's just about accumulating vulnerability. It's a very good point. Yes, ma'am.
So with the part in the next slide that cancer cells are constantly dividing and accumulate new mutations,
Yep.
you can have a tumor that develops because of a
Yes.
join one of those others. So that's how cancers progressively get
More aggressive, yes, yes, exactly. So they can start with one set of mutations and they can accumulate more progressively. Now, the accumulation of mutations doesn't have to make it increase in aggressiveness, but it certainly can change the how fast it spreads. It can change how to what extent it spreads. Right? It all depends on what the nature of the mutations are. Yeah. Yes, ma'am. No, that's an excellent question. So the question is, do benign tumors have all of the characteristics except for the last one? No. Benign tumors are very well-behaved tumors. Okay, they really are. They really are. And I think I'm not going to get to that slide in this go around, but I want to show you this one. Right? This is a nice picture that side by side that talks about that illustrates a benign tumor compared to a malignant tumor. Right? So what a benign tumor a benign tumor is usually going to be caused by some temporary inappropriate signal that cause the overgrowth of cells, right? So the benign tumor is the normal response to some signal that was inappropriate. Now, those cells that are produced, they are identical to the normal, they conform in terms of looking like the normal tissue, right? They notice that they're displacing the tissue around them, right? They're not injuring the tissue around them. The benign tumor is often well encapsulated, okay? And it's not growing in size, okay? So benign tumors are sometimes very mysterious as to why they develop in the first place, but they are very, very well behaved, right? And they don't have that sort of instability and those other features of cancer cells. Yeah.